Hello everyone, my name is Yutong. Today I'll be introducing to you an important property of genome graph that is called genome graph expressiveness and how we use it to improve genomes to genomes comparison. When we collect tumor samples from a patient, we often observe a diverse population of cells, and this phenomenon is called sample heterogeneity. Sample heterogeneity is not only observed in tumor analysis, but also in metagenome and immune repertoire analyses. In these cases, the sample collected same location will evolve over time and the composition of the cell population will change. This evolution often results in resistance or evasion of the treatment. It is then important to keep track of the evolution and be able to compare heterogeneous samples. The differences in cell populations can often due to mutations in their genomes. To model a set of heterogeneous genomes, we can use the heterogeneous string set to do that. And heterogeneous string set contains pairs of strings and their relative abundance, which describes the composition of the cell population. So when we want to obtain a sample distance, we can use the distance between the heterogeneous genomes to represent the differences between the samples. However, in real cases, we do not usually observe the complete genomic sequences. In fact, what we have due to limitations in sequencing technologies are fragmented sequencing reads. The fragmentation introduces another layer of abstraction and ambiguity, which prevents us from directly comparing them. The challenges introduced by sequencing reads can be alleviated by using genome graphs to represent them. Genome graphs have been used for decades to reconstruct and represent original genomes from sequencing reads. After genome graphs are constructed from two sets of sequencing reads that we're comparing, the problem of heterogeneous string comparison now becomes the genome graph comparison problem. The genome graph discussed in the scope of this presentation will have a source and a sink node, where each node and node label correspond to a substring from the genomes that we're interested in, and an edge will represent the adjacency between them, and there is a weight associated with it that represents the relative abundance of such substrings. And the string can be spelled out by following a path from source to sink by concatenating node labels on the path. Each path will use up the same weights for each edge on the path, and the weight will be the relative abundance of the string in the string set. Here we are assuming that a genome graph is a valid flow graph, which means for each node over here, the sum of weights on the incoming edges is equal to the sum of outgoing edge weights. This assumption is commonly applied in transcriptome assembly problems, and the similar assumption would apply to pangenome graphs that store DNA strings. And we say that a genome graph represents a heterogeneous string set if this string set can be reconstructed by spelling a flow decomposition of G. In other words, if we can find a decomposition of G into paths such that each path can spell out one string from a heterogeneous string set with equal weight, we say that a genome graph can represent this heterogeneous string set. The previous definition creates a correspondence between genome graph and a string set that it encodes. However, this correspondence is not one-to-one. In fact, for most genome graphs, there will be multiple flow decompositions so that a genome graph is encoding, instead of one string set, a collection of string sets, which we call the string set universe. And the capability of a genome graph to represent a diverse string set is the genome graph expressiveness. This complexity of genome graph, or the expressiveness of it, is studied in both the context of assembly graphs or in the context of pangenome graphs. And usually these bubble structures that you see where the edges branching out from a node and coming into another node is representing variations among the genomes that are storing in the genome graph. So due to genome graph expressiveness, there is a discrepancy between genome graph and the string set of interest. When a genome graph is constructed based on sequencing reads, it indeed represents the string set of interest. However, that string set is hidden in a string set universe that is encoded by the genome graph. So recall our original problem where we want to compute the distance between the genome set. Now we only observe the genome graphs that is a different representation. And the question we want to ask or answer is how accurately can genome graph distance estimate genome set distance? In our study, we aim to answer this problem by characterizing genome graph expressiveness, and we take a step further to reduce the discrepancy between the two representations. We first properly define the distance metric between heterogeneous string sets. We point out that in order to compare heterogeneous string set, we are essentially comparing two distributions of strings. Therefore, it is natural to use the earth mover's added distance or the wash assigned distance to compute the total amount of work that we need to transform one string set into another. In other words, we want to find a many-to-many mapping between a string set, and each edge in the mapping will have the cost of added distance between the two strings that are connected, and this distance will be weighted by the amount of strings we want to transform. And then the EMED, or the earth mover's added distance between two string sets, will be the cost of the minimum cost mapping between the string sets. As for genome graph comparison problems, there are previous work that proposed methods or definitions for the distance between genome graphs. And among them, GTED is an elegant metric that is applicable to general genome graphs because it tolerates repeats in the genome represented and assumes no knowledge on the complete genomes, which is applicable to our case. GTED, or graph traversal added distance, computes Eulerian tours in genome graphs such that the strings spelled out by the Eulerian tours are closest together. In other words, it performs alignment between strings spelled out in the Eulerian tour. One caveat about this distance metric is that it assumes the genome graphs are designed for storing and representing single genome by traversing Eulerian tours in them. And as a result, if a genome graph is actually representing multiple strings, an Eulerian tour will spell out a concatenation of these strings. And therefore, we might be performing alignment between the prefix of a string to a suffix of a string. And this will result in inaccuracies in distance estimation because we're performing alignments that will be otherwise impossible when we're computing the EMED. To account for this limitation of GTED, we propose flow GTED to account for the limitation of GTED, we propose flow GTED. We show that flow GTED can be defined using a similar definition as GTED, in that it finds a flow decomposition in each graph that minimizes EMED between them. To better understand this definition, let's first look at G2 over here. It is obvious that it has only one flow decomposition as shown in orange. And then according to the definition of FGTED, it will find the red decomposition instead of the gray one that we saw earlier, because now both red and orange decompositions spell out the same string set, and the EMED between them is 0. Recall that each genome graph encodes a string set universe, and the strings of interest is hidden in them. When we say that flow GTED minimizes EMED between encoded string sets, it is not only telling us the difference between the genome graphs, but also telling us the distance between the two items from each string set universe that are closest together. And since EMED also computes the distance between the string sets, we now are able to characterize the relationship between them because they're operating on the items from the same set. In theorem 1, we show that given genome graphs G1 and G2 and the string sets from their string set universes, the difference between EMED and FGTED is always greater than or equal to 0. Here we define the deviation as EMED between the string sets minus FGTED between genome graphs. With the lower bound, we saw how useful the string set universe is when we want to compare the two distance metrics. We took a step further and properly quantified the genome graph expressiveness. When we study the complexity or the expressiveness of the genome graph, we care about how different are the strings encoded by the graph. So we propose a metric called string set universe diameter that measures the difference between two furthest string sets from each string set universe. And we show that using string set universe diameters, or SUDs, we're able to upper bound the deviation by the sum of SUDs. Now we have a theoretical upper and lower bound for the deviation between EMED and FGTED. In the next part, we'll show how we can empirically estimate the deviation using genome graph expressiveness and use that to improve FGTED. We first visualize the discrepancy between FGTED and EMED on actual DNA sequences. We simulated T-cell receptor sequence set pairs, where each set has 2 to 5 strings, and each string is about 300 base pair long. We created partial order genome graphs based on the multiple sequence alignment within each string set. In this figure, we show the relationship between FGTED and EMED, where each dot here is a pair of sequences and genome graphs corresponding to each set. And the red line here shows the equality between FGTED and EMED. As expected, all FGTED are smaller than or equal to EMED. In order to estimate the deviation, we need to first quantify genome graph expressiveness practically. We do that by sampling flow decompositions in order to compute the sampled SUD. Recall that each element in the sample string set universe is coming from spelling a flow decomposition of a genome graph. Therefore, we define the sampled SUDs as a maximum distance between the two string sets that are spelled from sampled flow decomposition of a genome graph. We tested the sample string universe diameters on two types of graph. There is the multiple sequence alignment graph that we saw earlier, and we also constructed the de Bruijn graphs based on each sequence set. The de Bruijn graph is commonly used as an assembly graph to reconstruct the complete genomes from sequencing reads. The two graphs visualized here are actually constructed based on the same set of sequences, and you can already see that the structures of these two graphs are very different. For the MSA graph, the structure is simpler with a few bubble structure or the branching out structures that will lead to graph expressiveness. However, for the de Bruijn graph, it is way more tangled and there are way more bubble structures. And therefore, intuitively, we would think that the MSA graph will have a smaller string set universe and therefore a smaller diameter. And for de Bruijn graphs, there will be a larger string set universe and a larger diameter. We constructed two types of graph for each sequence set that we simulated. And here it shows that the distribution of diameter of each graph type. From this plot, we can tell that the diameters of the de Bruijn graphs are way larger than the diameters of the MSA graph. This validates our thought earlier that the MSA graph has smaller string set universes. Next, we compare the discrepancy between FGTED and EMED on both types of graphs on 225 sequence set pairs. And as seen from this plot for the de Bruijn graphs, the discrepancy between FGTED and EMED is larger because the blue points are further away from the red line that marks equality between the two distance metrics. Using the sampled SUDs, we visualize the relationship between genome graph expressiveness and the deviation. As shown here, each dot again is one of 225 sequence set pairs and a genome graph constructed on them. And there is clearly a positive correlation between sampled SUDs and their deviation. And in order to describe this correlation, we fit a line with a linear regression model. This linear regression model assumes that we know exactly what the genomic sequences are for each pair of genome graphs. And using the linear regression model, we're able to predict deviations in a pair of genome graphs where we do not know the complete genomes. We introduced this corrected FGTED metric by adding a correction factor, which is the predicted deviation on FGTED between two genome graphs. Here, the predicted deviation is produced by the linear regression model that is trained on 50% of data assuming knowledge of genomic sequences. And then the result visualized here are genome graphs where we assume that we do not know the exact genomic sequences. And as you can see here, that we successfully increased FGTED so that it is closer to the equality line as shown here in gray. We further validated our linear regression model on two data sets, that's the T0 sequences and the hepatitis B virus genome sets. Hepatitis virus genome sets are larger at 3,000 base pairs per string. And we perform a tenfold cross-validation to examine the average deviation of the corrected and uncorrected FGTED from EMED. During the cross-validation, we split 90% of data for training the linear regression model and the rest 10% for testing. And as shown in this table, the average deviation has been reduced significantly for all types of graphs and all data sets. Here we show that partly assuming that we have the knowledge of the genomic sequences can help us empirically improve genomes to genomes comparison, even though we only have the genome graphs. In summary, theoretically, we characterize the effect of genome graph expressiveness on genome comparisons. We defined string set universe diameters and use that to lower and upper bound the deviation of FGTED from EMED. And then practically, we take a step further to estimate the genome graph expressiveness via sampling and use that sampled SUDs to correct genome graph distance so that we can get a more accurate distance estimation between unobserved complete genomic strings using genome graphs. As a future direction, I would like to point out that the string set universe diameters are not only useful in genome graph comparisons, but also can be used as a regularization term for genome graph construction. There, the string set universe diameter can be used to control the variability of the string set universes, such that the genome graph can represent the reference pangenome more precisely. And therefore, it will be more accurate when it is used to do sequence to graph alignment or variant calling. With that, I would like to thank everyone in my research group for their support on this project. I would also like to thank my funding resources and thank you all for listening.